Hello. Everyone probably knows that blood cells are an important part of immune system. These cells help fight infections by attacking bacteria, viruses, and germs that invade the body. In line with this, our project is focused on blood cell subtypes classification. The LIFEBLOOD team members are Dichai, Willisini, Matawaran, Joseph, and Zida, Lunis. In this slide, we can see the outline of the presentation, which are introduction, objective, dataset, pre-processing of the data, defining functions, results, and conclusion. Introduction. Blood is a significant fluid that delivers important substances such as oxygen and nutrients for human beings, and it has a vital role in this time of COVID-19 pandemic since it supplies white blood cells to boost the immune system to fight for the virus and other diseases. However, an excessive amount of white blood cells can indicate that the body is infected by a disease, particularly pneumonia, urinary tract infection, leukemia, etc., while having an insufficient number of white blood cells can indicate that the bone marrow is failing due to a diseased liver. White blood cells are categorized into 6 different subtypes such as eosinophil, lymphocyte, monocyte, neutrophil, mononuclear, basophil plus lymphocyte and monocyte, and polynuclear, neutrophil plus eosinophil, and the number of each subtype in the complete blood count gives more details about the underlying problem. Therefore, monitoring white blood cells can help to reduce and prevent major health problems, and automating the classification of the images of the white blood cell subtypes through creating a convolutional neural network will be helpful in terms of time saving and improvement of clinical efficiency. Our main objective is to classify the images into different subtypes of white blood cells using convolutional neural network. The subtypes are basophil, lymphocyte, monocyte, neutrophil, and eosinophil. They can mononuclear, which is composed of basophil, lymphocyte, and monocyte, and polynuclear, which is composed of neutrophil and eosinophil. An important problem in blood diagnostics is classifying different types of blood cells. Here we have 410 original images and 12,500 augmented images of blood cells paired with subtype labels basophil, eosinophil, lymphocyte, monocyte, and neutrophil. We want to automatically classify each image according to the subtype of cells within it. The first approach will be to classify individual blood cells. Here we can see that with the original images we have imbalanced class sizes. Such imbalance can lead to misclassification of the model and lower the accuracy indeed. The basophil will be removed from our study because of the lack of samples. Now, we load training and testing data that consist the augmented images of 4 subtypes of blood cells such as neutrophil, eosinophil, monocyte and lymphocyte, with 2 cell groups as mononuclear and polynuclear. These are the samples of the augmented images of the following classes: eosinophil, lymphocyte, monocyte, and neutrophil. The augmented set have the same number of samples for each class. This balance will help our model on the tanning session to clearly recognize each class. Plus, the different view angles will help for generalization. Not much pre-processing is required for this dataset. Only normalization will be applied to reduce the pixel intensity range. Model summary. The model used here is a convolutional neural network which specs are displayed. The metric, optimizer and loss function are respectively accuracy, delta and categorical cross-entropy. Image data augmentation is applied to further expand the size of the training dataset by creating modified versions of images in the set. Callbacks function interact with the training model to save metrics after each epoch. These 2 plots attest that our model was able to learn useful features from our inputs. After 30 epochs, the accuracy reached 84% and a loss dropped to 37%. No over or underfitting can be observed. On the test set classification report, monocyte and lymphocyte classes have the highest score, around 90%, while the neutrophil and eosinophil are around 70%. In average, this model have an accuracy of 84% for all classes. The confusion confirmed what saw previously on the classification report. A certain number of neutrophil was misclassified as eosinophil. Some eosinophil was misclassified as neutrophil mostly. Monocyte was equally misclassified as neutrophil and eosinophil. All the lymphocyte were correctly classified. In this second part we will classify the two cells group mononuclear and polynuclear using the same model. After testing this model with binary classification, we found that the mononuclear has highest score of 100% and the polynuclear is around 89%. In average of this model also have an high accuracy of 94% for two classes. From the plot between accuracy and the number of epochs, it can be seen that the higher the number of epochs, the greater the accuracy until the 20th epoch onwards gives the highest accuracy around 98%. These two plots showed that the accuracy reach around 94% after 30 epochs and loss fluctuates after 10 epochs. In confusion matrix, we noticed that polynuclear was almost completely classified and some of mononuclear was misclassified as polynuclear. The accuracy of the model for the four categories is quite satisfying. The model performance are consistent on both training and testing sets. We can see this model reasonably worked on two categories with 94% as compared to 84% accuracy of four categories. Conclusions. White cells are involved in multiple medical procedures. Some require white cells type recognition. A model such this one can help the medical forces by automating this recognition process so that they don't have to spend much time on this task and focus on what matter the most, providing care to the people. Here are our references for this project. Thank you for watching our presentation.